So we moved from the nucleosome discoverer to the nucleosome structure solver. So Karolin has never been at the OMB actually. Her connection to Aaron is through Tim Bridgman, who was a postdoc with Aaron Klug, and Karolin was a postdoc with Tim. So technically she's his scientific granddaughter. Karolin did her PhD at the Biocentrum in Basel before moving to Tim Bridgman's lab at the ETH, where she was a key player in the efforts of solving the structure of very early nucleosome three-dimensional structures using X-ray crystallography. So after that, she moved to the US to start her independent career, first at the Colorado State University, and then recently she moved to UC Boulder, just across the state. And her lab not only works on the nucleosome and chromatin structures, but also now on how they assemble and recognize, as well as the evolutionary path of these nucleosome structures. And for those who know Karolin on Twitter, she's also a nucleosome police. Yes, go Karolin. She's won many awards. She's a HHMI investigator, and as well as a National Academy member, AAAS member, and an EMBO member. So without further ado, I will pass it to Karolin, and thank you for being here with us. And yeah, looking forward to your talk.
Well, thank you very much. Can you guys hear me okay? I hope that's okay. I'm really honored to be included in this honoring of one of the grandfathers of the nucleosome. And as was pointed out, I consider myself a second-generation chromatin structure biologist through my connection with Tim Bridgman. And I heard a lot about Tim's time in Aaron's lab and the coffee clutches that happened there. And hopefully sometime I can visit the LMB. It's never worked out for me. And in fact, right now, half of my lab is down with COVID. So it's probably good that I didn't come, because I might have been stuck there. So I thought today, because we are looking into the origins of nucleosome structure determination, at least parts of this symposium, to also look at the evolutionary origin of the nucleosome. But before I do this, I just want to point out that pretty much a lot of things that we now know about the nucleosome, we've already known in the 70s, thanks to work mainly from Aaron's lab. For example, it was first proposed that nucleosomal arrays, beads on a string arrays, fold up into these solenoid structures. This turned out to be possibly an in vitro artifact, but they can definitely be reconstituted at high resolution. We've known through work from Roger, of course, the histone composition of these arrays and the repeat length. And Aaron has also proposed a massive deformation of the nucleosomal DNA that turned out to be not as kinky as it was predicted, but still very, very profound. And then, of course, the seminal paper in 1977, based on a lot of biochemical data, cross-linking data, and X-ray diffraction data, as well as negative stain, resulted in a model of the nucleosome that is very scarily correct. And if you superimpose it with a modern day nucleosome structures, as you see here, to all practical purposes, all the information was already there. And I do hope Daniela will, because she's one of the pioneers of this work, will talk a lot more about how that all came to be. But it took a number of years, as you all know, until 1984 to arrive at a 7 angstrom crystal structure of the nucleosome. And then it took us another 13 years beyond that to obtain atomic 2.8 angstrom structure resolution. Of course, it's not atomic, but it's enough to resolve side chains and protein DNA interactions. Now, how we got from here to there is a whole different talk, and one that I've given often, and you can actually look it up in iBiology. I've done a little tutorial on the whole history and the technical problems that we had to overcome to get this accomplished. And this was, of course, a huge team effort in Tim's lab, and couldn't have been done without these amazing folks there. So the cryo-EM resolution revolution has not escaped the nucleosome, and in fact, nucleosomes are very, very easy targets because they're very recognizable. And I just want to point out that now, literally undergrads in my lab can get 2.5 angstrom structures of nucleosomes on a Titan Krios. But I will point out that this electron micrograph that you see here looks very similar in quality to the micrographs that were obtained in 1977. And you have to wonder whether if the equipment had been up to speed, we might have been able to pass around the whole pain and effort in getting a crystal structure of these particles and go directly from cryo-EM to a high resolution structure. So this was my little historical retrospective from my vantage point, and I do apologize. I know many people's contributions and seminal papers were not listed here. This was not meant to be comprehensive, but the reason why everybody was chasing the nucleosome like the dog after the proverbial bone is that they're very fundamental organizing principles of eukaryotic DNA. Because of the profound compaction, the wrapping of the DNA around the histone octamer, they really determine access to the genome, and that they profoundly affect replication and, of course, transcription, and require very, very complex regulatory machinery and ATP using machinery to pry the DNA off of the dead cold hands of the histones to be able to work on it. And this invariably holds true for all eukaryotes. And so I've become increasingly interested in the evolutionary origin of this packaging principle. And what you would normally do is you would look at the evolutionary tree and you would go to some lowly organisms, for example, Giardia. And it turns out all these nucleosomes look very much the same. Including all the machinery that's required to access the DNA. And this is because the histones, the protein components, are amongst the most conserved eukaryotic proteins known. And so we had to kind of go outside of the domain and take a look at Archaea. And John Reeve has discovered in 1990 that Archaea, almost the entire domain of Archaea, encodes minimalist histones that are undiversified. They're not clearly recognizable as H2A, H2B, H3, and H4. And this discovery really gave rise or helped to give rise to this idea that Archaea were the origins of the eukaryotic nucleus. And so we were wondering, what can Archaea tell us about the origins of the nucleosome? So unlike the eukaryotic histones, as I told you, they're not diversified. They usually just encode one or two that are very similar. They form homodimers or quasi-homodimers. They're very short. They don't have any of the external histone tails and the signaling functions that we have. And across the board, they're about 30-ish% similar as far as the histone fold is concerned. So the organization, so we determined a crystal structure of these histones bound to 90 base pairs of DNA. And this was a very difficult structure to determine, even by nucleosome standards. And I can go into details if anybody's interested. But once we solved the structure, we could see that the DNA organization of these archaeal histone dimers, homodimers, is very, very similar to what we see in the eukaryotic counterpart. And this holds true even to the minute details of the protein DNA interactions, the histone-like signatures that we see, that we know are characteristic of eukaryotic histones. We already see those in the archaeal histones. And similarly, the way by which two histone dimers join together in these four-helix bundle-like structures is also very similar. So the molecular details of the interactions, very, very similar. And so it's not surprising that we can actually assemble these histones into particles that look surprisingly and eerily similar to eukaryotic nucleosomes. And so Archaea have already invented this distortion of the DNA, but rather than forming discrete particles, these particles just keep going in the crystal lattice, forming these so-called continuous or endless nucleosomes, almost like a slinky-like spiral. So this was very intriguing, and we published this in 2017. But we next wanted to know whether, of course, this is a crystal lattice, do these slinky-like structures exist in solution and in the cell? And to look at that, we reconstituted these histones onto longer pieces of DNA, 207 base pairs of DNA. In theory, you have space for 7 histone dimers, so that's 3 more than you would get in a defined eukaryotic nucleosome particles. This is not the best image here. We were training on our F20 instrument that's quite old and long in the tooth and was supposed to be a little training program, but we got surprisingly intriguing 2D class averages, a minor class, these open particles here, and then a class that could best be fit with a closed nucleosome-like structure. So Sam Bowerman in my lab went ahead and determined the structure to low resolution of these open structures, and these can be fit with 4 histone dimers, and then the DNA veers off into an angle to bind an additional 3 dimers with the DNA. So all of the 7 dimers and the entire DNA is accounted for in these opened up, quite stable structures that you can see here. Just to make sure that we don't see like partial nucleosomes kind of sticking on top of each other. This is a flare of the electron density to prove to you that the DNA is really continuous and connected in these admittedly very low resolution images. What about the second class? This second class can be fit with 5 histone dimers and with 150 base pairs of DNA. So we have 1 additional dimer bound, allowing us to say that these form supernucleosomal structures that continue in their wrap. But then there's 2 dimers and 60 base pairs are missing in these rather defined structures. And so I forced Sam to sit down and take another hard look at the 2D class averages. And if you do the same, you'll see there's like this faint shadow of density kind of extending, almost like a hanger, closed hanger, extending from these more defined particles here. And with a lot of trying and noodling around with the data, Sam managed to actually build additional density for the DNA at very low resolution because it's very disordered to then demonstrate that now we have a 5+2 opening of these archaeosome structures. So what I've shown you is very, very consistent with what we've and others have observed in vivo. When you look at these bugs, these archaea, and you digest a chromatin in old-fashioned manner with micrococcal nuclease, you get a very profound 30 base pair ladder. And that's very different to the defined 150 or so base pair ladder that we observe in all eukaryotic chromatin. And this is very consistent with a slinky-like structure that opens stochastically and then permits access of the micrococcal nuclease enzyme to cut stochastically, to give rise to cut basically between every single histone dimer on average. So what have we learned from this? We have a system here in Archaea where we have already developed histones that bend and contort DNA in this profound shape that we know and love from eukaryotic histones. These histones are simple. There's only 1 or 2 of them. They don't have histone tails. They form, rather than defined particle, a semi-stable superhelix. And because this ability to stochastically open, the polymerase is slowed but not inhibited. And so there's no evidence for any of the regulatory apparatus that we have developed in eukaryotes. And these structures are also self-assembling. So then making the jump to eukaryotes and arguably allowing us this massive and obscene expansion of our genome size, we had to go to 4 histones with tails that allowed us to incorporate histone variants. And this in turn allowed us to build discrete stable particles from 2 different building blocks. And then in addition, have this graft onto these structures, this whole regulatory machinery of histone post-translational modifications, ATP-dependent remodeling factors, histone chaperones, and all these things that make studying transcription in eukaryotes so much fun. And so these eukaryotic particles now profoundly inhibit DNA-dependent processes, and they heavily rely on PTMs, on ATP-dependent remodelers. But as I told you, even the most primitive eukaryote, primitive in quotation mark, has this entire machinery already completely assembled. And so we're still looking for the missing link. How did we get from the left-hand side to the right-hand side? And because I'm a microbiologist by training, I'm really intrigued by weird organisms. And we happened onto giant viruses. These viruses are large DNA viruses, and they mostly infect amoeba. The last thing we need is more viruses, so fear not, these affect amoeba. And they are huge, they're almost as large as bacteria. And the reason why they are so large is because it lacks the intricate receptor uptake mechanisms that modern viruses have, and they basically just pretend to be bacteria. The amoeba eats them, and then they die a horrible death after that, and the virus multiplies in them. Why are we interested in these viruses? There's a subclass of these viruses that have very large genomes. They replicate exclusively in the cytoplasm of the host, and they encode histone-like proteins. And so we thought this was something that we could contribute to. Intriguingly, these histones are very evolutionary, very distant, and they're also very old if you do some phylogeny. But the most fun aspect of these histones is that they are fused. So H2B is fused as one polypeptide chain with H2A, and H4 is fused to H3. And unlike the archaeal histones, they are clearly recognizable as such. And so they have all the additional trimmings that, for example, make H3 characteristically. H3 is this N-terminal helix here, for example, and the viral histone H3 will also have this additional alpha helix here, and so on. So we decided we'll just express these histones, we put them onto DNA, and we'll see what we can see. Can we make nucleosomes? So the questions we asked is, can they bind DNA? Can they form nucleosomes? If yes, what properties do these nucleosomes have? When, how do they arrive at the viral factory? And I think most importantly, as a biologist, we'd want to know, does the virus need its own histones? And if yes, what does it need it for? So to answer the first question, we made these nucleosomes. This was actually surprisingly hard because they're not very stable, but we managed to obtain very beautiful 2D class averages and then managed to solve the structure on 2 different lengths of DNA, shown here on 147 base pairs of DNA and here on 207 base pairs of DNA to resolution that is sufficient to build a model into these structures. Now, what do we see? We can actually fit these structures with homology models of the histone doublets that we can make and minimize. And so they fit actually quite well. And to all practical purposes, these structures do look like nucleosomes. If you are used to looking at nucleosomes, you might notice that they are a little more bloated, meaning that the distance between the DNA gyres is a little bit widened because they're destable, and that results in their destabilization. If you superimpose the histones, just half of the nucleosome in this case here, with the eukaryotic histones in gray, you will see that it's quite good superposition. All of the trimmings are there, all of the secondary structural elements are there, and they pretty much do the same thing. But the biggest difference is that the DNA, only about 110 or so base pairs of the DNA are organized. And in fact, not only that, but the DNA extends in a very defined manner. Usually when we don't see the DNA, it's because it's too disordered and it's averaged on the Cryo-EM grid. In this case, they are actually quite rigidly arranged, and we don't really know what causes this arrangement. The other difference is that these viral nucleosomes are much less positively charged than the eukaryotic counterparts. And again, they have to be a little more destabilized because arguably as soon as the virus gets expelled into the cytoplasm or gets eaten by the amoeba, it has to shed the histones in order to allow transcription to take place. So the overall positive charge is quite a bit lower. And that's particularly obvious in the so-called dyad region. This is the region that is very, very stably bound by histones H3 and H4 in eukaryotes. And there's much less positive charge here, resulting in a destabilized particle. And you'll also note in this comparison eukaryotic nucleosomes with the viral nucleosomes, they're a little bit more bloated and splayed apart. What about the connectors? This is unique to the viral nucleosomes. H2B is quite easily connected with H2A. The density isn't very good, and there's a number of connectors that are in molecular dynamics simulations compatible with the disordered density that we see in this area. So it's quite easy to loop from the C terminus of H2B to the N terminus of H2A because they're very close together. The situation is quite different for H4, H3. The H4 C terminus is at the inside, in the inside of the nucleosome. And the N terminus of H3, where it has to connect to, is way here at the other side. And so what happens is that this tail loops over, and the connector weasels its way between the two gyres of the DNA superhelix. As you can see here, it's like in between. The sequence of these connecting sequences is quite conserved between different viruses in this class, and the density is actually good enough to see a number of interactions, hydrophobic interactions with the histone core, and also interactions with the DNA. But this is why the ends of the DNA are probably disordered. It just gets pushed out of the way a little more, and that's also why these particles are a little bit more bloated. And again, molecular dynamics simulations show that there's a number of slightly different configurations that are compatible with the energetics and with the density of this region. And I will say that we've done more molecular dynamics than we would normally do because of COVID, because this is something you can actually do when the lab is shut down. So a lot of people in my lab turned to be quite enthusiastic modelers. All right, but I think most importantly, what do these histones do in the virus? And so we teamed up with Chantal Abergel in France, who's an expert in these giant viruses, and we wanted to know how do these histones know where to go? Because remember, the way this virus replicates itself is it gets digested in the cytoplasm, and it never enters the nucleus. Rather, it kind of pokes holes into the nucleus. It compromises the nucleus so that the transcription machinery leaks out and takes over the job of transcribing the viral and transcribing and replicating the viral genome. So when we take GFP-tagged amoeba histones and inject them into the amoeba, and transiently transfect them into amoeba, they know exactly where to go. They go immediately to the nucleus shown here, and they stay in the nucleus even after we've infected with a virus, presumably in getting incorporated into chromatin. When we do the same experiment with GFP-tagged doublet viral histones, these guys have no idea where to go. So they just kind of hang out in the cytoplasm. Some go in the nucleus, probably by mistake. But when we infect with a virus and the viral factory starts to form in the cytoplasm, they massively accumulate in the viral factory and congregate there. And we can observe this for histones H2B, H2A, and also GFP-tagged H4, H3. And indeed, they do co-localize in these viral factories. When you take infectious, healthy fat viruses and do quantitative mass spectrometry, you will note that they have massive amounts of histones. In fact, there's more H4, H3 than there's major capsid protein. And histone H2B, H2A doublet is not really far behind. And when you do some fancy back of the envelope math, you can calculate that there's enough viral histones to package the entire viral genome into nucleosomes. So the most important question, I think, is, are histones needed for virus fitness and infectivity? And so just to reiterate the life cycle of this really interesting virus, it gets eaten by an amoeba. It sheds, presumably sheds the histone, so the DNA is out in the open. It somehow compromises the nucleus so that the transcription machinery and replication machinery leaks out. Histone genes are transcribed, they're translated into histones, but rather than going to the amoeba nucleus, they stay in the cytoplasm and go back into the viral factory where they are assembled into infectious virus particles. The amoeba bursts, literally bursts, and new Melbourne virus can go and infect new amoeba. So when you make a virus without H4 and H3, and this was a really difficult experiment to do because this system is not, to this until now has not been genetically accessible. So Hugo Bisio in Chantal's lab worked very hard to make this happen. The viruses actually, when you knock out H4, H3, they assemble, but they are not infectious. And we in fact believe that they don't have any DNA in them. The amoeba lives until you transiently transfect the amoeba with viral H4, H3, in which case it will happily make infectious virus, because now it can reassemble its genome into nucleosomes. And so this really shows that these histones are essential for viral fitness. So we've answered some questions. We've gotten more, as any good science project should. They can form nucleosomes. These nucleosomes are destabilized and bloated. The connectors and the tails have structural rather than regulatory functions, but we're still working on identifying possible post-translational modifications on these. We don't really know how they know to go to the viral factory. The most boring scenario would be that they're attracted by the massive amounts of viral DNA. We don't know how they're assembled in a stepwise manner, which has to happen for eukaryotic nucleosomes. H3, H4 has to go on first, followed by H2, H2B. And these histones are required for viral fitness and for infectivity, but why? We don't really know. It could be as boring as just making sure that you can package the entire large genome into the capsid, but we actually believe it has additional functions that we're really curious about. So I want to end by acknowledging the people in my lab, my Nucleosome Evo group, Young Lu, Chelsea, and Cody, and especially my collaborators, John Reeve, the Sant'Angelo lab, and then the Albergel lab, who've been really a lot of fun to work with. And I'm happy to answer some questions. So feel free. I don't bite. I'm on Zoom, so feel free to answer to ask questions. Thank you very much.
Thank you for a great talk. So I'm Alex Bateman from who does the PFAM database. So I'm really interested in families and evolution. I was wondering if you'd looked in the Asgard phylum.
Yes.
Archaeobacterium, because I note there a quick look, you can see things like histone deacetylase homologs. So maybe they're a bit more eukaryotic-like.
Yes, we are actually working on the Asgard nucleosomes as well. Some of them have tails, which makes them particularly interesting. We don't have anything to talk about quite yet. And of course, the huge downside of the Asgard is that nobody can grow these things because they live in families with a lot of other organisms, and they're very hard, or if not impossible, to cultivate.
Mark?
The giant viruses are amazing, but do you think it more likely that they picked up the genes for these histones from their amoeba hosts rather than being a missing link?
Yeah, we don't know. It's debatable. I think that's the more parsimonious explanation, that they picked them up from some ancient amoeba host. It is known. People have done phylogenetic analysis, and it's kind of a little above my pay grade. I don't really understand how you arrive at these conclusions, but the acknowledged conclusion is that they are ancient. And so if they pick them up from somebody, that's somebody that eukaryote was an old eukaryote. But then there's also a field, or some folks believe that possibly the virus was the original donator of the histones to the eukaryotes.
Maybe I missed it. I have a quick question. So how many other families of viruses that have histones? Do you know?
So yeah, so there was a recent meeting on the evolution of chromatin that I went to, and there was one talk, somebody who looked through a lot of virus families and found quite a few additional ones that have histones. And so I think this is kind of something that's now people are paying attention. is something that's really coming more to the forefront. And in fact, it also appears that not quite a few bacteria also have histones that look exactly like histones.
Cool. Okay, if there's no more question, thank you very much for the nice story, and hopefully next time you can visit us in person. Thanks everyone for attending, and we'll be back at 3:30 for tea. So thanks, Karolin.